Okay, so I'm going to talk about my work, which is building these models of a part of the motor cortex of the mice. And first I want to explain some of the motivation. So we have seen in the last years, 20, 30 years, like an explosion of experimental data. So we have data, especially for many parts of the brain, but for the cortical circuits, for the cortex, which is like the outer layer of the brain, where many of the high-level functions are located. We have seen a lot of data on, for example, the different cell types, the cell types that compose each of the layers of cortex. Also their connectivity, their physiological properties, so how these different cell types respond. Also like the inputs they get from different parts of the brain. So there's a lot of data from many different studies, and the idea is that we have to make sense of all this data. So one way to do this is to build computational models that try to integrate all these disparate studies into something coherent that you can play with. So we focus on primary motor cortex, M1, in mouse, and we work together with an experimentalist called Gordon Shepherd, which is the son of a very famous Gordon Shepherd in neuroscience. And he does a lot of these experiments in mice, both at the single cell level. So he records from single cells, measures all the physiological properties, and then also in terms of the connectivity. So he looks at how all these different cell types are connected, what inputs he arrive from different parts of the brain. And I'm going to give you a brief overview of how the motor cortex circuits are organized. So you essentially have three types of excitatory cell types. And this has been shown not only for motor cortex, but also for all the different cortical areas. So that's interesting because both the visual cortex, the motor cortex, somatosensory cortex, they all share these different three cell types. And these are called IT, which stands for intratelencephalic, which just sort of means those cells that project internally within cortex, and what's called the striatum, which is another part of the telencephalon. So you see here these IT cells. They are located in all of the layers of cortex. As well, I didn't mention that cortex is divided into 6 layers, from layer 1 to 3, 4, 5, which is divided into parts, and layer 6. And that's depending on the sort of cells that are located in each of the layers. Okay? So these IT cells are located in all 6 layers. Well, sorry, not except one. In layer 1, there's usually not many cells, just inhibitory cells, but in all the other layers. Then you have the PT cells, which are called pyramidal tract cells, and they are the ones that sort of project out of cortex down to spinal cord. So they're very important because they're sort of the output of the circuit. They imagine motor cortex is the part of the brain that controls your movement. So these are the cells that project from the cortex down to the spinal cord to send the signals to move. And then we have this third type of cells, the CT cells or corticothalamic, which project back to thalamus. So thalamus is an important region we were talking about before. It provides sort of inputs from the external world into the brain, and these are cells that project back. So they sort of form a loop between what's happening in the cortex and the inputs to it. So you see in the diagram, again, that these IT cells are located across all layers. The PT cells are only in layer 5B, so they're very specific to this part of cortex. And the CT cells are only in layer 6. Okay. So one very important feature of motor cortex that was discovered is that these IT cells project to PT cells, but not the other way around. So you have a sort of downstream connection where these IT cells are supposed to be processing information within the brain. So for example, communicating with the sensory cortex areas, integrating information from what we are seeing, what we are feeling, and processing this information, and then sending the motor commands down to PT cells, and then PT cells down to brainstem. Okay, so it's a sort of two-step process where ITs process information, they send to PT, and then to spinal cord. So you see in the diagram here, a very important connectivity feature is that we don't have these connections back from PT to IT, only that way. Also shown here is this H current modulation, which is one of the channels that PT cells have. It's called the HCN channel, and it has lots of functions. It has been shown to be involved in many different processes, but one of the one we're interested here is hypothesized to be sort of a switch for PT cells. So it can turn the output of PT cells on or off, and there is a way to modulate this HCN channel with neuromodulators from other parts of the brain, norepinephrine, for example. So it's a way of changing from a state of imagining or planning what you want to do. So if I want to start walking now, first I have to think about it. There's a stage of planning, and at that stage I'm not really moving. My muscles are still, but I'm still planning what I'm going to do. And then when I actually decide to make the movement, then I activate these PT cells and I start moving, so my muscles get activated. So this HCN current is one of the hypothesized mechanisms to change from motor planning to motor execution. Yes?
Does it mean that you can modulate the H current in a time of milliseconds, hundreds of milliseconds, moving from planning to execution?
I believe so, yes. The norepinephrine effect is quite fast, as far as I know. Okay, so based on all this, yes? Okay, just stretching. Okay, so based on all this experimental data that we have, we built this model of M1 microcircuits. So here in the left, I'm showing sort of the region that I'm simulating. So it's a sort of cylinder, like a column, in the M1 cortex of the mouse. And this column has approximately 10,000 neurons. So in real size, so the real scale of the brain, this region of cortex, which is about 300 micrometer of diameter, and the whole cortical depth, which is like 1.3 millimeters, you can fit there 10,000 neurons and about 30 million synapses. Okay, so that's the region that we're simulating. And you can see here a sort of representation of how the network looks, although this is a sort of downscale version with just a few of the cells to show how it would look. And within this region, we have tried to map as closely as possible to biology using experimental data. So for example, we have, for each of the layers in the cortex, we have the density of neurons that you would find in cortex. So for example, layer 2 is 110,000 neurons per millimeter squared. Okay? In the layer 6, it's a bit more, so there's a higher density of neurons. So yes.
Millimeter cube?
Millimeter cubes, yes. And then for each of the layers, we also know the composition. What cells are present in each layer? So as we saw before, in layers 2, 3, 4, 5, and 6, we have these PT cells, which are represented by the red cells. In layer 5, we have the PT cells, which is the blue cell here. And in layer 6, we have the CT cells, the corticothalamic. Okay. So notice that we have different models. So for the IT5A, the layer 5A IT cell, and the layer 5B PT cell, we modeled it in a lot of detail. I'm going to show more about that. For the other ones, we modeled them with more simpler models, like this one have 5 compartments. So you were seeing before yesterday how to build cells of different resolutions. And although they're a bit simpler, they actually reproduce also the physiology. So we optimize these cell models to reproduce the spiking properties of the different cell types in different layers. And then we also have 2 types of interneurons, the low threshold spiking and the fast spiking interneuron. And those are present in all layers, which is also found experimentally. So just to illustrate the complexity of this, as we were showing, Michele Migliore was showing how to optimize cells. So we did that for one of the corticospinal cells. So here's some data, real data of how the cell looks with some imaging data. And this is the simulation reconstruction. So you see that it's basically identical. We use 700 compartments to reproduce very accurately the shape of the neuron. And we also reproduce very accurately the response of the neuron. So we see here the response to different current inputs. And also this thing that is called the FI curve. So the firing rate of the neuron in response to different current injections of different amplitudes. And you see that it matches very nicely the response of the original neuron. And to obtain this tuning, what we did was modify the parameters of the cell, like you were doing yesterday with the KM channel. So we're playing with all these different channels, different calcium channels, sodium, potassium, but we did this automatically. So we ran what is called evolutionary algorithms on supercomputers. So this took about a month on a supercomputer. I don't know exactly the number, but thousands of cores, of computation cores, to obtain this model that is very accurate. So the other big thing that we really wanted to incorporate is all this idea of connectivity. And connectivity, you can think of it in different scales. So first you can think about the connections from other parts of the brain into this circuit, into the M1 cortex. So for example, for that we used data from one of Gordon's papers, where he's showing the inputs from different parts, for example, thalamus or M2, which is the secondary motor cortex, or S1, which is the somatosensory cortex, where these inputs connect to, and those are very specific. So for example, the S1, they project mostly to the neurons in the layer 2. The M2 inputs project mostly to the neurons in the layer 5B. Okay, so it's a very specific connectivity pattern that we incorporate here. We also look obviously at the local connectivity. So how are the cells in this part of the circuit that we're simulating, how do they connect to each other? And that is also very specific, and we have data for like each of the cell types, and depending on the depth of the cell, so not just the layer they belong to, but also the cortical depth of the cell. So if you think about this neuron, the PT cells, they are in layer 5B, but layer 5B is super wide. So what we found actually experimentally is that the connections from the layer 2-3 cells to the PT cells in layer 5B are much stronger at the top of the layer. Okay? So it's not just connectivity based on the layer, but also on the cortical depth within that layer, sublaminar level connectivity. And finally, we also incorporated information about the distribution of inputs. So today you were locating one of these synapses in the dendrite, but imagine if you have a neuron with 700 compartments. You need to know where these different synapses go. And so Gordon Shepherd also had information about, for each of these projections, so for example, the inputs from thalamus to the layer 5 PT cells, what part of the cell do they target? Okay? And you have these kind of density maps, which shows you where most of the synapses are located for that specific projection, from thalamus to PT cells. And you have that for each pair of connections in the model. So from layer 2-3 to PT cells is a different distribution of synapses. So we incorporated also all that information into the model. So this is a summary of the connectivity of the model, where you can see these lines on the left are the inputs from other regions. So these are two parts of thalamus, S1 and S2, other parts of motor cortex, and something called orbital cortex. And then the different populations in the different layers. And you see basically the pattern I was talking about before. So you have this recurrent connectivity at the top, which is sort of cells communicating with each other and across cortex to process information about the sensory inputs. And then this very strong connection coming from layer 2-3 to the PT cells, and particularly to the upper half of layer 5B. And remember these PT cells, the blue ones, are then projecting down to spinal cord. Okay, so yeah, just remember this is a summary of the connections, but in reality the connectivity depends on the exact cortical depth of the cell. And this is an example of this distribution of synapses that I was talking about. So we have here two different maps. These are inputs from thalamus to PT cells. And these are a representation of how the final density of synapses looks in the actual model. So you see the brighter colors show a higher number of synapses. And the same for inputs from S2. So S2 to PT cells, you see the distribution of synapses is quite different. So this is also interesting because it means that different inputs project to different parts of the cell. And as we were saying before, there are lots of effects of these dendritic computations. So the fact that, for example, they are in different parts of the dendrite means that you can integrate information from these two regions. So the PT cells are combining information from thalamus and from S2, for example. Okay, so to simulate this model, which has 10,000 neurons, some of them very detailed, of course you require a lot of computational power. So one simulated second of the model required about 1 hour on 50 cores of the supercomputer. That's the whole 10,000 neurons with 30 million synapses. Of course, when you have this type of models, you also need to optimize the parameters of the network. So you need to run the network many different times with different parameters to make sure that you reproduce more or less the experimental data that you have. And then you also want to do experiments. So you have your model and you want to try different stimulation here and there. So essentially you need to run this model for many, many times. So obviously we started with this university cluster that had 500 cores, but it very quickly became obsolete. So we didn't have enough power to run what we needed. So we moved to something that are scientific shared clusters that you can run simulations, but you have to wait on a queue for a long time. So that was very frustrating. And finally, we have settled with Google Cloud. So the university afforded to pay for it. And so we're able to run on up to 50,000 cores at the same time. So it was great because like in a couple hours, we can run thousands of these simulations, explore lots of parameters, and then get the results and analyze them. And before we took maybe a week to do that. So just quickly some of the results that we obtained. So this is a raster plot of the network. So you can see all the different layers and the spiking of the neurons. So each dot is a spike, and this axis here is the time. So you can see some interesting things that the firing properties depends a lot on the cell type, on the layer. Even within the same layer, you see different spiking properties here in PT cells, for example. So we match these average firing properties and other properties of the firing patterns to experimental data. And we show here it's consistent with what you see in mouse. Interestingly, there was a paper when we finished the model. There was a paper showing that there is a big difference in the function of these different layers of PT cells. So the upper cells apparently are more involved in planning, and the lower ones in motor execution, which was something that sort of consistent with what we were seeing in the model. We also looked at LFP oscillations. So LFP is local field potential, and it's something that you can obtain by introducing an electrode in sort of the extracellular part of the brain and measuring the voltage there. And it really shows the activity of thousands of neurons, potentially, that are around that electrode. So it's a measure of combined population activity. Yeah?
How do you implement that in simulation?
So, you can... Oh. So you can basically calculate it from the currents of the neurons. You don't have to do it manually because it's implemented in this tool, but you're essentially calculating the currents produced by each segment of the neurons, and then calculating the distance from that segment of the neuron to the electrode, and you have a formula, and you get this LFP signal. So it's in that
sort of like, can you implement an extracellular electrode in the model?
Yes. So you simulate. You can put the electrode at any location in your circuit and calculate the signal from there.
Well,
and
but that's not quite in the circuit, right? It's kind of like outside the circuit.
Well, yeah, in the tissue that we're simulating, some of the tissue is neurons, and then you have sort of extracellular space. So this is built into the tool, so you can build any model that you want and just say, I want the extracellular LFP in this location, and you get it automatically. So this was interesting because we didn't really enforce any oscillation activity, but from the model we saw these emerging oscillations in the delta. Delta is about 0 to 4 hertz, and the beta gamma range between 25 and 40 hertz. And that is actually what you observe also in the real cortex. And we didn't really put any inputs that were oscillatory into the network. So these properties we assume emerge from the sort of the connectivity properties, the physiology of the neurons, sort of naturally from the model. And we also saw this phenomenon called phase amplitude coupling, where some of the oscillations vary their phase the amplitude of the fast oscillating gamma frequency here, you see depends on the phase of the slow oscillation. So this is between 0 and 4 Hertz, and this is the gamma, very fast, and the amplitude is modulated by this phase. And apparently this has also been shown experimentally. This in prefrontal cortex, for example, there is then delta gamma coupling, and it has been linked to rolling movement in motor cortex as well. So this is the spectrogram where you also see that it's, over time it changes the frequency that you have. So there's a bunch of interesting things that we can look at now in the model. And related to what the talk this morning, we also look at this idea of information flow. Although this Granger, we use a measure called Granger causality, which is a bit controversial, and we are not so happy about it either. But it did show kind of some of the features that we know about motor cortex. For example, this thing I was talking about before, where IT cells only project to PT cells, but not the other direction. And yeah, so we compared a few things as well, looking at the oscillations, the frequencies where most of the information is transferred. So that is also consistent with these gamma oscillations we were seeing. And because we have the model, now we can sort of play with it. So we want to see what happens when you provide inputs from thalamus, from this part of the brain called thalamus. So we can increase the inputs from that particular part and see how the dynamics of the network are modified. So what gets activated, what populations fire, which ones don't. And so we see, for example, when you activate the thalamus, this goes mostly to the upper layers, so you get a lot of activity here in layer 2-3, and then some activity in the PT cells, because these are connected from layer 2-3 to PT. However, if you activate this motor cortex, secondary motor cortex, which projects directly to this layer 5B, then you get a stronger activation in the layer 5B region directly without going through the upper layers. So this, for example, would be a way of these higher motor areas producing some output movement directly without going through sensory cortex. And finally, we can evaluate sort of molecular or pharmacological effects. Because we have such a detailed model with all these ionic channels, we can try different things. So we, in particular, wanted to look at this effect of the HCN channel, IH. And this is just showing that our cells reproduce what we see experimentally. So if you have these HCN blockers, you see that the response of the PT cells increases. So this is the normal condition. And when you block HCN, so the response of the neuron increases. And when you do this in the circuit, what you get is the mechanism I was talking about before, whereby you can modulate and go from a planning or preparatory state, where the IH is high, to an execution state where the IH is low. So this would be sort of supporting the theory that the IH can be involved in this sort of activity. And something that I mentioned the other day, I think, is that when you build these models, you have to do sort of statistical analysis. So you cannot just build one network, but you have to build many with some randomization. So you change how the connections are created, and you change the sort of inputs that you give it, all within the parameters of your model. And you can obtain, for example, 25 different networks, and then compare these effects across the networks. And if you get something that's statistically significant, you can say that your model in general actually has that effect. Okay, so if you find it in just one of the 25, that is not such a strong proof. Okay, so yeah, in summary, the M1 model, also NetBind, which is the tool that I'm going to talk about now, is useful as a sort of theoretical framework to integrate all of this experimental data that you have. So to make sense of 30 or 40 different studies, it's nice that you can put it all together into a model and then play around with it. So in this M1 model, we have data from over 30 studies. So then you can do things like study the dynamics, how the different cells are activated, information flow, these multiscale interactions where you change molecular effects and see what happens in the network dynamics. And in theory, you can develop hypotheses that can then be tested experimentally. So with Gordon Shepherd, we work closely together. So now we're looking at, for example, the interaction between different inputs, between thalamus and S2, for example. And now, in theory, he would build some experiments to test whether our model predictions are correct. And then we can also go back to the model and fix whatever he finds. So we have like a sort of loop between experiment and modeling. And of course, ultimately, what we want to do is understand diseases better and try to develop treatments. So with these models, you can simulate some disease and maybe try out some experimental pharmacological treatment or neurostimulation, and without harming any patients, of course. And that's it. Any questions? Yes.
How do you have the input to the model? Is it like a bunch of random stimulations somewhere?
Almost, but not really. So what we do is, these are the actual inputs from other parts of the brain. So you see here we have like 7 different parts of the brain that provide inputs to that circuit. So this is what is really driving the model, driving the network. So the activity in each of these regions is random, as you're saying, but it's very connected in very specific patterns. So you see here, this one's project to the top, which that's strength, this one to the bottom. So it's sort of random, but the connectivity that is driving the network is sort of biologically based. So something we want to do next is build models of all these different parts, so we can connect all these regions together.
You have about 10 questions. I'll focus on one. It's more about cerebellum oscillations and theta frequency that you did not mention in there and then specifies. But the most important question, well, two. So I saw that you already applied this model to control a robotic arm. I saw a publication.
It was a previous version of the model, like a simpler version. Yeah.
Okay. And yeah, this one looks more complete. But especially 1 month ago, Walter Sam came to Zurich to present his theory about how the different neurons interact in the cortex and how they relay information and match it against the prediction. And he has this design of theoretical design because it doesn't have a simulation. So he came to us to ask if we could implement these simulations. And actually, you already have everything implemented. So I understand that you can run these simulations with very fast supercomputers, but do you have a mean to simplify also the network so that it could be run on a faster system?
You mean simplify the model itself, so you can?
Yeah, with neurons. And did you consider this idea of learning? Because you didn't mention any plasticity in this.
No, that's something we want to add, but we don't have plasticity yet.
But there are a lot of data on that, so
yes,
it would be possible for you, especially because you have all the tools to tune the parameters of plasticity.
Yeah. So the previous model you're mentioning, the one that controls the arm, that had plasticity. It had the spike time dependent plasticity and reinforcement learning. Yeah. But we haven't yet done it here. So this is like the first version. We just finished the whole thing, and now it's like lots of things we want to do. But
did you consider the use of generalized integrated fire neurons to substitute these complex models?
So we, in general, in the lab are more interested in the detailed version, but we know that it can be useful to have like a simpler version. And of course, as soon as we publish, we provide all the connectivity, so it should be easy to build like a model. Also, within NetPine, you can very easily change the cell types. So you can change it for generalized integrate or Zikevich.
10,000 neurons, you can actually move directly to one of those neuromorphic chips you saw yesterday.
Yeah.
And they need the connectivity because they don't have any information about that.
Yeah, for sure, that's an option. Okay. So
do you
now,
want to take another break or you want to go forward with that? I think I'll
forward.
Go. But I ran away.
Yes, yes, you ran away.
But maybe tomorrow I can ask more questions.
Yes. So, or whatever you want.